Imagine a world where we can cure genetic diseases, grow crops resistant to pests, and even produce life-saving medicines, all by rewriting the very code of life itself. Sounds like science fiction, right? But this is not the distant future, it's happening right now. Thanks to recombinant DNA technology. This groundbreaking method has transformed medicine, agriculture, and scientific research, giving humanity the power to solve problems that once seemed impossible. But how does it work? And why is it so revolutionary? Let's dive into the fascinating world of recombinant DNA technology. Recombinant DNA technology is the process of combining genetic material from different organisms to create a new customized DNA sequence. This technology allows scientists to isolate specific genes from one organism and insert them into another, granting new traits or abilities. The DNA that has been altered is called recombinant DNA, and the organisms carrying this new genetic material are known as genetically modified organisms or GMOs. This technology is the backbone of modern genetic engineering, allowing us to cross the boundaries of nature by merging genes from different species. How exactly do scientists create genetically modified organism or GMOs? The process involves 5 main stages: isolation, cutting, insertion and ligation, transformation, and expression. 1. Isolation stage. In this step, scientists identify and extract the gene of interest from a donor cell. This gene contains the instructions for a specific trait we want to transfer. At the same time, a small circular DNA molecule called a plasmid is extracted from a bacterial cell. Why a plasmid? Because it can replicate itself and is the perfect vessel to carry the gene of interest into another organism. 2. Cutting stage. Once the gene of interest and plasmid are isolated, they must be cut using special enzymes called restriction enzymes. These enzymes act like molecular scissors, slicing DNA at specific points. The cuts create sticky ends overhanging sequences that make it easier to attach new DNA. Both the gene of interest and the plasmid are cut to prepare for the next stage. Comprehension check. How do restriction enzymes help in the process? It acts as in molecular scissors, cutting the DNA in a specific point. 3. Insertion and ligation stage. Now, it's time to combine the pieces. The sticky ends of the gene of interest and the plasmid fit together like puzzle pieces. To seal the bond, scientists use an enzyme called DNA ligase. This enzyme works like molecular glue, permanently joining the DNA fragments. The result? A recombinant plasmid, a hybrid DNA molecule containing genetic information from 2 different organisms. Comprehension check. What role does DNA ligase play in recombinant DNA technology? Its purpose is to act as a molecular glue joining the 2 DNA fragments together. Transformation stage. In this critical step, the recombinant plasmid is introduced back into a living bacterial cell. This process, known as transformation, allows the bacteria to absorb the modified DNA and begin replicating it. The bacterial cell now holds the blueprint to express the new gene. 5. Expression stage. Finally, as the bacteria reproduce, they make copies of the recombinant plasmid and begin expressing the gene of interest. This step is called gene cloning. Comprehension check. What are the 5 main stages of recombinant DNA technology? It's isolation, cutting, insertion and ligation, transformation, and expression. So, how does this technology impact our lives? The possibilities are endless. Let's explore some real-world examples. In the past, Insulin was extracted from the pancreas of animals such as cows and pigs. This process was expensive, time-consuming, and not always effective. Today, genetically modified bacteria can produce human insulin, making treatment for diabetes more affordable and accessible. Bt corn in agriculture. Before genetic modification, farmers relied heavily on chemical pesticides to protect corn crops from pests like the corn borer. Recombinant DNA technology solved this problem by creating Bt corn. Bt corn is a genetically modified crop that contains a bacterial gene for insect resistance. This reduces the need for chemical pesticides while increasing yield. In the past, clotting factors needed for hemophilia treatment were extracted from human donors, which posed risks of contamination and was costly. Recombinant DNA technology solved this using sheep. Now, transgenic sheep have been engineered to produce clotting factors in their milk, offering a safer and more efficient way to help people with blood disorders. Genetically modified yeast producing vaccines. Previously, vaccines required the use of live or weakened viruses, which carried risks and were difficult to produce in large quantities. Today, genetically modified yeast cells can mass-produce vaccine components, such as the hepatitis B vaccine, making vaccination safer and more effective. Finally, golden rice for vitamin A deficiency. Many people in developing countries suffer from vitamin A deficiency, leading to blindness and immune system problems. In the past, supplementation programs were the only solution, but they were not always accessible. Now, golden rice, a genetically modified rice variety enriched with vitamin A, helps combat this deficiency and improve public health. Comprehension check. Can you name at least one real-world applications of recombinant DNA technology? While recombinant DNA technology holds incredible promise, it also raises ethical questions. Should we modify the genetic code of living beings? What are the potential risks of releasing genetically modified organisms into the environment? Could this technology be misused? As science advances, society must carefully weigh the benefits against these concerns. With responsible innovation, recombinant DNA technology could continue to unlock new solutions for global challenges. Recombinant DNA technology is not just a scientific breakthrough, it's a gateway to shaping the future. By understanding how to manipulate DNA, we hold the power to cure diseases, fight hunger, and protect our planet. And who knows? Maybe one day, you could be the scientist who changes the world through genetic engineering. Final question. Based on the animated module you watch, what is recombinant DNA technology and what it is purpose, and why it is important? While recombinant DNA technology holds incredible promise, it also raises ethical questions. Should we modify the genetic code of living beings? What are the potential risks of releasing genetically modified organisms into the environment? Could this technology be misused? As science advances, society must carefully weigh the benefits against these concerns. With responsible innovation, recombinant DNA technology could continue to unlock new solutions for global challenges. Recombinant DNA technology is not just a scientific breakthrough, it's a gateway to shaping the future. By understanding how to manipulate DNA, we hold the power to cure diseases, fight hunger, and protect our planet. And who knows? Maybe one day, you could be the scientist who changes the world through genetic engineering. Now that you know what recombinant DNA technology is, answer the following question. 1. What is the main purpose of recombinant DNA technology? A. To create identical twins. B. To combine genes from different organisms. C. To make artificial intelligence smarter. D. To change the color of human eyes. 2. What enzyme is used to cut DNA at specific sites? A. DNA polymerase. B. RNA ligase. C. Restriction enzyme. D. Helicase. 3. Which of the following is an example of a GMO? A. Normally born sheep. B. A dog born with no tail. C. Bt corn. D. Twin babies. 4. What is the role of a plasmid in recombinant DNA technology? A. It acts as a carrier for the gene of interest. B. It breaks down proteins. C. It provides energy to the cell. D. It destroys foreign DNA. 5. Which of the following is not an application of recombinant DNA technology? A. Insulin production. B. Golden rice. C. Human cloning. D. Bt corn. 6. What enzyme acts as molecular glue during DNA recombination? A. Restriction enzyme. B. DNA ligase. C. RNA polymerase. D. Helicase. 7. In the past, how was insulin obtained for diabetic patients? A. Extracted from cow and pig pancreas. B. Synthesized in plants. C. Created using artificial intelligence. D. Harvested from human donors. 8. What does gene cloning involve? A. Making copies of recombinant DNA. B. Cloning an entire human. C. Erasing unwanted genes. D. Changing the gender of an organism. 9. What is the main benefit of genetically modified yeast for vaccines? A. Reducing costs and increasing production efficiency. B. Eliminating the need for medical professionals. C. Making vaccines taste better. D. Replacing all natural immune responses. 10. Which crop was modified with a bacterial gene to resist pests? A. Wheat. B. Bt corn. C. Soybeans. D. Rice.